Thank you for allowing me to present my lab's work on trying to discover new causes, genetic causes of hereditary breast cancer. So just a little bit of background about the genetics of hereditary breast cancer. There are a number of genes that cause breast cancer, and these range from high penetrance genes such as BRCA1 and p53, which are very high penetrance but are very rare in the community. And they then also range these common genetic variants, these SNPs, which each confer a lower risk of breast cancer, but are very, very common, and there are lots of them. So my lab's been trying to discover the full repertoire of these genetic causes of breast cancer. And the reason we're doing that is because a large proportion of women who are diagnosed with a breast cancer and attend a familial cancer centre and have a genetic test don't get any definitive genetic result. So all known genes are sequenced and no mutation is found. And so why is it important to identify the cause of their breast cancer? Well, it's a couple of main reasons for that. Firstly, if you don't know what the specific gene mutation or the gene is, you can't give proper primary and secondary cancer prevention management. Because if you don't know who's a carrier and who's not, all family members, all blood relatives need to be treated as if they had equal risk, which obviously they don't. In addition, particular genes will predispose to particular cancers, and so that can inform what type of screening would be necessary for that individual. In addition, for treatment of the cancer, it's well known that some these genes will make that cancer susceptible to particular treatments. So, for example, a BRCA1 or BRCA2 tumour are more susceptible to treatment with platinum-based drugs or PARP inhibitors. So, for those reasons, it's important to be able to offer a genetic cause for all women who are diagnosed with a hereditary breast cancer. So at least for more than 10, 15 years, our laboratory has been trying to uncover this missing heritability, and we've been doing that through case control studies, in the BEACCON case control study here, where we've accumulated over 6,000 cases of women who are diagnosed with breast or ovarian cancer, but have tested negative for all known genes. And then we've compared that to the sequencing data from women from the LIPAL cohort who are controls, who are healthy women attending breast screen. And again, we have about 6,000 women here. And this work was done, I say, over about 15 years or so in 3 phases of sequencing. And this is just really to show the history of this. We started off very early on when exome sequencing was first developed. We sequenced 150 cases from 69 families, identified genes, and then we sequenced those candidate genes, up to 1,400 of them, in 2,000 case controls. And then as sequencing got cheaper, we did another 3, almost 4,000 case controls. So overall, we have data for 6,000 cases and 6,000 controls. And if you're interested in seeing the details of that study, it's been published in NPJ Breast Cancer. So what did we find from that? How successful were we at finding new genes? Well, we were able to, first of all, confirm known genes, so CHEK2, PALB2, and ATM, were all mutations were in excess in the cases versus the controls. But the critical thing to note here is that the number of families explained by each of these genes was very, very small. So just looking at these loss of function mutations, PALB2, less than 1%, CHEK2, 1.3%. And so overall, each of these genes didn't really explain many of the families. But we were interested in new genes. But when we looked at the new genes as well, this is some of them, but we had a bigger list of these. But even if all these genes are real, most of them, again, only were present in less than 1% of the families. So even if all of our candidate genes that had an excess of mutations in the case controls was real, we would still only be explaining maybe 10% or also of that 6,000 families that we were sequencing. So that was sort of informative, but it was disappointing that we really thought that this would have solved a much larger proportion. So the question was, where is the missing heritability? So our data certainly shows that there are many genes that can cause breast cancer, but they each contribute to a very small number of families and certainly can't explain all the families or even many of the families. One possibility is that we've just picked the wrong genes and we've done something wrong. But in one sense, it's pleasing that other investigators internationally and big groups as well have also failed to identify any new genes. And really, since the discovery of BRCA1 and 2 back in the 90s, PALB2, which accounts for about 1% of breast cancers, has been really the only major new discovery in hereditary breast cancer. So it's not just us. There's something going on with the genetic causes of breast cancer. So one possibility is that there actually aren't any new genes and that most of the families are caused by known genes, but we're looking in the wrong place. We're not looking at mechanisms that are detected using clinical or laboratory-based tests. And one of those is obviously non-coding variants. These haven't been looked at much because in general it's been too expensive looking at the non-coding regions, but certainly it's a possibility. But I want to talk today about promoter hypermethylation. Again, a very plausible mechanism because we know in primary cancers, in primary breast cancers, genes such as BRCA1 and RAD51C, for example, they do get methylated in the tumors. So it can occur somatically, so it's plausible that could also occur in the germ cells and be inherited. And indeed, there is some evidence for this mechanism occurring in cancers and breast cancer. And so the first report was of inherited methylation in the gene MLH1 in Lynch syndrome. So a small number of these families, you could detect methylation of this gene in blood DNA. At 50% of the DNA was methylated at this gene. And then there's been one study of the BRCA1 gene in 49 index cases from hereditary breast and ovarian cancer families. And this group, headed by Gareth Evans in Manchester, identified 2 of those families which had methylation in blood DNA. And this was 50% of that DNA had methylation. So clearly a constituent of methylation. However, they're not the only 2 studies, but there have been lots of other studies. Most of them had limitations. And firstly, the sample sizes were very small because the methods are very expensive and tedious. So the studies have been small. Many of the studies, a vast majority, didn't use hereditary cancers. They were looking at sporadic cancers. So their chances of finding a hereditary cause of breast cancer in a sporadic case is going to be much, much lower. But the biggest problem is that most of them use technology that just wasn't really up to scratch. So they used array-based technologies, which are kind of expensive, a bit difficult to analyze, and can only give a broad overview of the methylation, and they can't get down to base level data. Other assays that are PCR-based are very, very accurate, but only assay a few CpGs. You can see just a very, very small window of the promoter of the gene. And it's also, these assays are very tedious to set up, and so you can only ever screen a small number of samples. So our hypothesis was, and in fact, promoter hypermethylation might explain a sizable proportion of hereditary breast cancers, and the only reason we haven't discovered it is because there wasn't any sort of comprehensive next-generation sequencing-based technology which could analyze large number of samples in a comprehensive way. So that's why we collaborated with Twist and were able to get early access to their next generation sequencing methylation detection system. And in this system, you enzymatically convert the DNA from the C's to a T's, and Twist developed probes depending on if the DNA is methylated or unmethylated. They make probes that are going to be specific depending on which outcome there might be. So there's a mixture of probes there that will pull out methylated sequence and unmethylated sequence in an unbiased fashion. So when you sequence it, you can get a genuine image of what's actually happening in that DNA with respect to promoter methylation. So again, because the assay is so easy and relatively cheap, we were able to sequence the full promoter of 16 hereditary breast and ovarian cancer genes, so not just the CpG arm, but the whole promoter. And we're able to do this in 417 blood DNAs from index cases who were negative for all known breast cancer genes. And importantly, the average Manchester score, which is a score that indicates how likely it is that it has a genetic cause, was 26.9, the average, which is high. And we had 74 that had a Manchester score greater than 34, which is very high. But that's the level from the study by Gareth Evans. His 49 cases had Manchester scores greater than 34. So we really wanted to replicate the same sort of risk profile in this study. As I mentioned, none of these families had mutations in known genes, looking at the exons and the central splice site. In addition, we also sequenced DNA from 49 breast cancers from DNA extracted from formalin-fixed paraffin-embedded blocks, because we certainly want to look at that if we found any methylation in the blood DNA. Importantly, the twist only requires 200 nanograms of DNA, and certainly for some of the samples we had less than that, and it still seemed to work okay. We used Illumina MiSeq 150 base pair paired end reads, and we used Bismarck software for the analysis of the data. So here are the 16 genes that we looked at. So some of these are well-known breast cancer genes, others are not quite so well-known, but we included them because they are sometimes included on commercial hereditary breast and ovarian cancer panels. So here was the design. So the target size is about 68 kilobases, 16 regions, 16 genes, and there was 504 probes to cover those regions. 72 of the probes were removed due to repeats. And so overall, the covered size is 59.9. Now, so the overall, from what we wanted to target, there was 88% of that. But actually, this is pretty good because the promoters are highly repetitive, have a high GC content. And so because of that region, this is actually very good, and we're very satisfied with that. Often you find if you're trying to design a PCR assay for a promoter, you just can't do it because the sequence is so GC rich you can't do it. So the array-based approach actually seems very robust. So here's the overall coverage of the CpGs in the blood DNA and the FFPE tumors. I just need to note here, these numbers seem a little bit low, but this is the coverage based on the targeted region, not the actual design region. So, but essentially, if you just look at the region that was designed for, because some of the regions we couldn't target, these numbers are in about 85 and 88% for the 10x and 20x coverage. The medium depth was very good for the blood. We were aiming for about this, and we got it. The FFP tumors were a little bit lower, not unexpected because that DNA is often very, very degraded and relatively poor. Importantly, the cytosine to thymine conversion, because the Twist kit uses an enzymatic method, was very good. So for the majority of samples, we had 99% conversion. And the reason some of those weren't as high is because the DNA quality was quite poor, and surprisingly, some of the clinical samples, the quality there was poor because they've been sitting around for many years. But for all the high quality control samples, the conversion was 99.8% or higher. So all those technical aspects of the assay were all looking very, very good. So here's some of the germline results. So here are some heat map plots from the Bismarck. So here's the p53 gene, for example. On the bottom here is the samples. Here's the mean coverage. Here's the CpGs across the gene. And here's the UCSC designation of where the CpG islands are predicted to be. And you see here for the majority of these samples, there's no methylation going on in the blood, which is expected. And so that's pleasing to see that. One thing we didn't want to see is lots of red, sort of random patterns here. Now, importantly, we know the systems work because here we have a positive control. This is fully methylated DNA control. And you see here, all these are red, which is what you're expecting for 100% methylation. So this is all looking very good. But in terms of our study, none of these samples have methylation of p53. So just showing that's not a fluke, here's red 51C. Again, the positive control is looking really good. The samples that didn't work here, this one had very, very low coverage. So it looks like it's got methylation, but that data is not reliable. But for all the other samples, you can see the data looks very, very clean, no methylation. Again, PALB2, the same thing, control looks good. but no methylation. BRCA2 again looks very, very clean. And then finally on the BRCA1, which we were particularly interested because of the Gareth Evans work, again, we didn't have any methylation occurring in any sample. And the only time we did see something going on again for those few samples, the metrics were very poor, suggesting that they're failed samples because of DNA quality, but not because of the actual twist assay. So these results here is summarized very, very easily. We didn't find any constitutional methylation, i.e. 50% of the alleles methylated in any of the 16 genes in the 470 cases, and in particular, none of those that had very high Manchester score. So very, very quick result. But in fact, this really is more data that's been collected for hereditary breast cancer and methylation of hereditary cancer genes than's been generated in the last 10 years. So with this one assay, in a matter of a couple of months, we were able to really resolve this entire field. Hereditary methylation is not a major mechanism of hereditary breast cancer. So just wanted to touch on the FFP tumors because we will do some more samples and maybe some of them might have it, but it's not going to be very common at best. But I know many of you might be interested, can you use this method on tumors from FFP blocks? So we had 49 breast tumors that we dissected the DNA from these archival blocks. And the majority of these cases provided good data. And even if some of the data is a little bit noisy, you could certainly get readable data for the purpose that we were looking for. And so here's an example of some tumors down here. So the same sort of image as before. Here's the mean coverage. Here is the promoter region of P10 here. The majority of these tumors aren't showing any methylation in P10. But again, the control sample here showing full methylation. Again, the one sample here looking like it's got some methylation has very low coverage. So again, that one is a failed sample, again, because of the tumor quality. Again, here's BRCA2. Sometimes you get a sample that looks like something going on, but again, that's very low coverage. But the vast majority of samples work very well and clearly have no methylation going on. PALB2 was similar here. A couple of samples with a bit of methylation, but poor quality samples, so probably not real. But we get to BRCA1 here, and here we have the control sample showing good methylation across the gene. But here we have a sample here which has methylation across the entire promoter region. The coverage is really good. The conversion rate is very good for this sample. And here's some data. This is sample 105. And this is actually triple negative breast cancer, which is consistent with loss of BRCA1. It's got LOH at chromosome 17, suggesting it's lost the wild type allele and it's retained the methylated allele. It has a high HRD score because we've sequenced the tumor by whole genome sequencing for this sample. And so it has lots of the rearrangements. So it's not lost its homologous recombination repair capacity and other metrics like mutation signatures. It looks very BRCA-like. So this clearly offers an explanation for why this sample is BRCA1-like. And so before we did this assay, we just knew that it was a BRCA-like tumor, but couldn't explain it. But with this assay, we were able to then give a molecular explanation for why that tumor looked like it was a BRCA1 germline tumor. So that was very pleasing. And again, this is just showing that same sample here plotted in a different way. Here's the mean CpG methylation across all the FFP samples. And here's this sample here showing about 60% of the DNA was methylated across that region, which matched that sample at about 60% purity of the tumor. Importantly for the FFP tumors, you are able to predict reasonably well which samples might work. So you can do an assay before you proceed to the to methylation sequencing. And this is simply a Van Beers PCR, which is a cocktail of primers that amplify different size fragments. And if you only amplify 200 base pair fragment, it's not going to work very well. Might still be okay, but not as well as if you can amplify much larger fragments. And the same thing here with the 10x coverage. 200 base pair kind of works okay, but much better to have a higher fragment size. So you could do this assay first, and you could then exclude these samples that you knew wouldn't work very well. Also, I didn't mention it from the other slide, but some of the samples were very new. We had about 20 of those, which were blocks that were generated only about 6 months ago. They all work very well. The blocks that didn't work so well were the much older blocks. So again, new samples from new paraffin blocks should work pretty well. So one of the other things that was important from using this twist assay that gave us an insight that wasn't possible prior to having this assay available is, what is a promoter? What is a promoter? Where should we be looking? Because most studies have just used PCR assays, and you had to take a guess at where the best region was. But here's the methylation profile across the region that we sequenced here. This is the promoter. This is the UCSC predicted CpG. And you see here, the vast majority of the bloods actually have methylation to a high extent. So this really can't be a genuine promoter methylation of BRCA1. What we preferred was looking at this region here. There's another small CpG island here. This seems to be far more predictive and reliable measure of the promoter methylation. And that information actually explains some of the studies that people who have looked at promoter methylation of BRCA1 in blood DNA using PCR-based assays, and the frequency that have been getting vary from like 8%, 40%, 47% here, 22%. Big differences in the frequency, and other studies that are not shown here were just a few percent. But when you look at where they were sequencing this region here. And it's no wonder they were getting a high proportion of those samples having a high frequency, a high level of promoter methylation. And really a better assay would have been something around about here. So this is information that you wouldn't have got without having the twist assay that enabled looking at the whole promoter. Now, we were looking for 50% level methylation, so a constitutive methylation of a fully inherited methylated allele, but we also had the ability to look at lower levels of methylation. So shown on here, we have the average methylation across the samples in the germline, and here's one of those samples pulled out randomly, showing that it sort of maps with the red sample here, maps with the blue control samples. But we did get some samples that seem to have a low level of methylation here. So the blue is the control, this sample here showing about maybe 10% of the allele of the CpGs are methylated, and a second sample here, maybe slightly higher level of methylation. So the assay, even though we weren't looking at it specifically, this is also an important area of research, because this level of methylation can indicate an increased risk of future breast cancer. Not as high as if this was fully knocking out the gene, but it's an important application of this twist assay. So what are the conclusions of this study? Well, there are technical conclusions. So first of all, we couldn't substantiate the data from Evans et al. That inherited methylation of BRCA1 is common. We didn't find any methylation of BRCA1 in 417 cases. So it's certainly not anywhere near. They had 2 out of 49 cases. But also, in addition to that, we're able to show that there's no methylation going on in any of the hereditary breast and ovarian cancer genes. So clearly, this is not a common mechanism for hereditary breast cancer. But however, we also are able to show that low-level promoter methylation is easily detectable, and is certainly another application for this technology for people who are looking for like a population-based assay to look for lower levels of future breast cancer risk. So the lessons learned here with respect to the twist assay. So the assay actually works very well. I was very surprised and very pleased at the performance. It works well on the blood DNA, and some of the samples didn't work so well, but they were poor quality DNA. Good quality DNA gave excellent performance, and particularly pleasing was the FFPE DNA, that they worked surprisingly well, given the fact that FFPE DNA is quite deranged and can use just as little as 200 nanograms of DNA. You can predict whether the FFP DNA is going to work with a simple assay. And overall, the assay was very simple, really no different from the scientist, from my perspective, to any targeted panel, apart from the extra step of the enzymatic conversion. So this is a key strength of this assay. It's very, very simple. And as finally, as that's mentioned in the last few slides, looking at the entire promoter really gives you critical insights into what's happening in that promoter. And it does explain some of the contradictory conclusions that different laboratories had, particularly for the BRCA1 promoter. And clearly this could also be happening for lots of other genes. So clearly this has lots of applications for not only for our work here, but for future studies of promoter methylation in cancer. So I just need to acknowledge the people who have contributed to this work, and particularly to the patients and families who contributed their DNA to the study. Thank you.